Observational. To appreciate how flowering plants reproduce, we have to look past the visible bloom and focus on the hidden machinery inside. The female reproductive core of the flower is the gynoecium. It consists of a pistil, which is divided into 3 distinct physical zones. The stigma at the top acts as a landing platform for pollen. The style forms an elongated tube, and the base widens into a bulge called the ovary. This fundamental architecture varies across plant species. A flower might possess a single pistil, making it monocarpellary, or it may be multicarpellary, producing several pistils at once. As you can see in these 3D models, multicarpellary flowers organize in one of 2 ways. The pistils can be fused together into a single unified structure, known as syncarpus, or they can remain completely free and independent, which is called apocarpus. The structure serves a dual mechanical purpose. It provides a targeted runway to capture pollen on the stigma, and a highly engineered protective fortress that funnels genetic material down into the hidden ovarian cavity below. Inside that basal bulge is the ovarian cavity, or locule. Here we find the placenta, which generates the megasporangia, more commonly known as ovules. Each ovule relies on a physical tethering system. It is anchored to the placenta by a short stalk called the funicle. The exact junction where the body of the ovule connects to this stalk is the hilum. To defend the inner cells, the ovule generates its own outer armor. 1 or 2 protective envelopes, called integuments, wrap almost entirely around the cellular mass inside. Looking at this cross-section of a typical ovule, notice the tip. The integuments do not close completely. They leave a tiny gap called the micropyle, establishing a future gateway for the pollen tube. Directly opposite this micropylar gateway is the chalaza, which forms the basal end of the ovule. This intricate system of stalks, shields, and gateways exists to protect the nucellus, a central mass of cells packed with food reserves. It is the staging ground for creating a new generation of life. Deep within the micropylar region of the nucellus, one specific cell differentiates itself. Packed with dense cytoplasm and a prominent nucleus, this is the Megaspore mother cell, or MMC. The MMC starts out as a diploid cell, meaning it carries 2 full sets of chromosomes. To prepare for sexual reproduction, that genetic material has to be reduced. The MMC achieves this through Megasporogenesis. It undergoes a meiotic division, physically splitting to cut its chromosome count in half. The immediate outcome of this division is a linear arrangement of 4 separate haploid megaspores. We see a biological rule of attrition common to flowering plants. 3 newly formed megaspores degenerate and die. Only 1 functional megaspore survives. Because the entire female gametophyte forms from this lone cell, the process is defined as Monosporic Development. By having the genetic material and sacrificing 3 out of 4 cells, the plant focuses all of its structural resources and energy into one functional champion. With the competition eliminated, the functional haploid megaspore begins a rapid internal expansion. The single nucleus divides mitotically, splitting into 2 that migrate to opposite poles. 2 more divisions follow. The 2 nuclei become 4, then 8. Crucially, these divisions are free nuclear. No cell walls are built between the newly formed nuclei. This strategy allows the plant to efficiently clone its genetic blueprint and position the nuclei in open space before expending the energy to construct permanent walls. Once the 8 nucleate stage is reached, the free nuclear phase ends. Cell walls are finally laid down, dividing the internal space to organize the mature female gametophyte. Looking at the mature embryo sac layout, 3 chalazal nuclei are walled off together, forming the antipodals. At the micropylar end, 3 more form the egg apparatus, a central egg cell flanked by 2 supporting synergids, which feature specialized basal thickenings called the Filiform apparatus. This structure serves as a localized chemical beacon, effectively guiding the incoming pollen tube exactly where it needs to enter. The remaining 2 polar nuclei are never walled off individually, floating together within the massive central cell. This creates a definitive count: 3 antipodals, the 3-celled egg apparatus, and 1 central cell. A mature embryo sac is a 7-celled structure, despite housing 8 nuclei. This microscopic 7-celled vault is the final destination. Hidden deep within the ovule, it waits precisely aligned for the exact moment of fertilization.